In 1952, Alfred Hershey and Martha Chase conducted a series of experiments in order to determine whether bacteriophage DNA, or its proteins, were the genetic material transmitted to phage progeny. In one experimental group, they tested the phage proteins, while in another, they tested its DNA. In the first step of the experiment, Hershey and Chase radioactively labeled one sample's protein capsules with sulfur-35, a radioactive isotope of sulfur. For the DNA sample, they labeled the phage's DNA cores with phosphorus-32, a radioactive isotope of phosphorus. Next, they infected E. coli bacteria with the radioactively labeled bacteriophages. Then, the mixture of E. coli cells and phages were placed in a blender to shear off the protein coats. Next, the mixture was centrifuged to separate the protein from the cells. When centrifuged, the bacteria forms a pellet at the bottom. Hershey and Chase then examined the progeny phages when these cells lysed. In the protein sample, the sulfur-35 was found in the supernatant fluid containing the viral protein coats and not in the pellet containing the bacteria or in the progeny, indicating that the protein was not passed on to the progeny and not the genetic material. In the DNA sample, the phosphorus-32 was found in the pellet and in the phage progeny, indicating that the DNA was the genetic material.